Have you ever wondered what it would be like if we could actually rewrite the very code of life itself? Well, today we're talking about a technology that's doing exactly that. It's called CRISPR. It's this absolutely revolutionary tool that's on the verge of changing medicine as we know it, giving us the power to edit our own DNA. Let's dive in.
So, I want you to think about your genome, your complete set of DNA, as this enormous instruction manual. And I mean enormous. It's 3 billion letters long, and it has all the information needed to build and operate, well, you. Ever since we finished the Human Genome Project, we've been able to read this book of life.
But, just like in any book, typos can happen. A single letter out of place, one tiny mistake in those 3 billion letters of code, and the result can be a really devastating genetic disease.
And we're not just talking about a handful of rare conditions here. Scientists have been able to compare the genomes of healthy people with those who have certain diseases, and they've pinpointed over 5,000 disorders that are caused by a single known genetic mistake. For thousands of these diseases, we know the exact cause, right down to the letter.
So that brings up this massive question, right? If we know exactly what the typo is, and we know what the correct spelling is supposed to be, what if we could actually go in there and fix it? What if we could finally move beyond just reading the book of life and actually start rewriting it? Now, you'd probably think a tool this powerful would have to come from some futuristic, high-tech lab built from the ground up. But the amazing thing is, the solution was actually found in one of the simplest forms of life on Earth, bacteria. We basically stumbled upon nature's very own incredibly precise gene editor. The whole story was like this decades-long scientific detective case. Way back in the 80s, scientists in Japan noticed these weird repetitive clusters of DNA in bacteria, but had no idea what they were for. Then, fast forward to the early 2000s, and a researcher named Francisco Mojica has this huge aha moment. He realizes that the DNA in between those repeats were perfect matches for the DNA of viruses. This couldn't be a coincidence. It had to be some kind of immune system. And that's exactly what it is. The name CRISPR, I know it's a total mouthful. It's just an acronym for how the DNA sequences look. But what it does is way more important. It basically acts like a genetic memory card for the bacteria, keeping a record of all the viruses that have tried to attack it in the past. So here's the play-by-play of this brilliant little search and destroy mission. A virus attacks, and the bacterium snatches a small piece of the virus's DNA and files it away in its CRISPR library. Now, if that same virus ever tries to attack again, the bacteria makes a copy of that stored DNA. It's called a guide RNA. This guide then joins up with a protein, a kind of molecular scissor called Cas9. Together, they hunt down the invading virus, find the matching DNA sequence, and just snip, threat neutralized. So this bacterial defense system is just ingenious, right? And it led scientists to ask this really groundbreaking question. If bacteria can program a protein to find and cut a specific piece of DNA, could we hijack that system? Could we use it to target and edit our own genes inside our own cells? And this was a total game changer, because you gotta understand the old gene editing tools were just incredibly clunky and difficult. For every single gene you wanted to edit, you had to build a whole new, really complex protein from scratch. It was like having to build a different phone just to run a different app. But CRISPR changed everything. With CRISPR, the Cas9 protein is the hardware. It's like the smartphone. And the guide RNA, that's the software. You just pop in a new guide RNA, which is super easy to make, and you can tell the exact same hardware to go to a completely different gene. Dr. Feng Zhang, one of the pioneers in this field, nailed it with this smartphone analogy. See, the real power isn't just in the cutting, it's in the programmability. And that is what made gene editing suddenly so much cheaper, faster, and accessible to thousands of labs all over the world. Okay, but Cas9 has a bit of a limitation. It's really, at its core, a pair of molecular scissors. Its main job is to cut right through both strands of DNA. Now that's great if you want to just disable a gene, but what if you need to do something more delicate? What if you need to fix a single letter typo without breaking the whole DNA strand in half? Well, that exact question led to the next generation of gene editors. We're talking about much more sophisticated tools that act less like scissors and more like the find and replace function in a word processor. This chart is a great way to see how these tools have evolved. You've got your original CRISPR-Cas9, the molecular scissors, perfect for cutting and breaking a gene. Then came base editing. Think of this one like a pencil and an eraser. It goes to a specific spot and chemically changes one DNA letter into another, all without breaking the strand. And then the newest tool in the box is prime editing. This is the full-on find and replace function. It can literally rewrite a stretch of DNA with new information, which lets it fix a way bigger range of mutations.
Okay, so this is where it all gets very, very real. This is not science fiction anymore. These tools are officially moving out of the lab and into the clinic. And they're producing these incredible, life-changing results for actual patients.
This is Alyssa. When she was just 13, she was diagnosed with T-cell leukemia. And after chemo and a bone marrow transplant both failed, her doctors told her family that, well, there were no other options. But then, she became the very first person in the world to be treated with a base-edited therapy. Scientists took immune cells from a healthy donor and used base editing to engineer them, turning them into cancer-hunting machines. And today, years later, her cancer is in complete remission. She's back in school, just living her life. It's unbelievable. And the speed here is just breathtaking. There was a baby, known as KJ, who was born with a really severe genetic disorder. Scientists were able to design, test, and deliver a personalized base editing therapy just for him. The whole process, from start to finish, something that would normally take years, was done in just 7 months. And the breakthroughs are just happening one after another. We've seen the first treatment where base editors were injected directly into the body, a single shot that permanently lowered dangerous cholesterol levels. We've seen the first time a disease-causing mutation was directly corrected inside a patient's body. And now, prime editing is already in clinical trials, just a few years after the science was first published. The momentum is just, wow. So where does it all go from here? The way we're doing things now is incredible, but it's very focused on creating one specific drug for one specific mutation. So what happens when a single disease can be caused by hundreds of different genetic typos? This is the huge challenge known as allelic diversity. Take a disease like cystic fibrosis, for instance. It can be caused by hundreds of different mistakes in the very same gene. Trying to develop and get approval for hundreds of different drugs, all for one disease, it's just not practical. And that is pushing scientists toward the next holy grail, so-called disease-agnostic therapies. The idea here is to create a single treatment that could work for lots of different mutations that cause the same disease. Or, maybe, even a single treatment that could fix multiple different diseases that all share a similar kind of genetic error. But as amazing as all these editing tools are, they all face one giant hurdle: delivery. The real bottleneck right now isn't the editor itself, that's the payload. As one expert put it, the biggest challenge is figuring out how to get these powerful little molecular machines into the exact right cells, in the right tissue, inside the human body to do their job. Solving that delivery problem, that's the next great frontier. This technology is fundamentally changing our relationship with disease itself. It brings up these profound questions about the future of medicine, about ethics, about what it even means to be human. It really forces us to stop and ask, how is our world going to change when a cure is no longer something we just hope to discover, but something we can actually sit down and write ourselves?